Imagine your immune system as this, like, highly fortified military base. And your dendritic cells, or DCs for short, they are the elite scouts.
Right. They're constantly out there.
Exactly. Just patrolling the perimeter, moving through your tissues, looking for any enemy invaders, things like sneaky viruses or rogue cancer cells.
Yeah, they are the absolute front line.
And for years, scientists thought they knew exactly what kind of fuel these elite scouts needed to sound the alarm when they finally found a threat. We really thought we had the blueprints to their engines completely figured out.
We really did. The dogma was incredibly rigid.
Right. But there's this groundbreaking May 2026 paper published in the journal Cell Metabolism by Horace Murillo and this huge international team of researchers, and it has just completely flipped the script.
It is a massive paradigm shift in how we understand our own defenses.
So, okay, let's unpack this. Because our mission for today's deep dive is to go completely microscopic. We're going to unpack every single conceivable detail of how mitochondrial metabolism directly regulates these frontline defenders.
Tracing the exact mechanism, yeah.
Right. From the flow of a single electron in a cell's power plant, all the way down to the physical locks placed on your DNA.
And to really grasp the scale of this, we have to look at the old dogma we mentioned.
Mm-hmm.
Previously, the consensus was that when these dendritic cells were activated, they relied almost entirely on glycolysis to get their energy.
Glycolysis being that, quick cytosolic fermentation process.
Exactly. Just quick energy. And on the flip side, oxidative phosphorylation, or OXPHOS, which is that slow, highly efficient energy-generating process inside the mitochondria, well, that was strictly associated with immune tolerance.
Meaning the field believed that heavy mitochondrial activity was basically the signal for the immune system to stand down.
Right, to suppress its response entirely.
Which is wild now that we know better.
So let's meet the main characters of this story. In the world of conventional dendritic cells, we primarily look at two subtypes, right? We have cDC1s and cDC2s. Yeah, and you can think of the cDC1s as your special operations forces. They absolutely excel at this really complex process called cross-presentation.
Right, this is where they capture pieces of a dead tumor cell, right?
Yes, they capture those pieces, the antigens, and present them to CD8 plus T cells. They're essentially priming those T cells to become intense, targeted killers against cancer and viruses.
Wow. And the cDC2s?
They are more like your standard infantry. They have different immune functions, primarily dealing with other types of extracellular pathogens.
So to understand how these scouts power those totally different jobs, we have to look under the hood at their engines. We mentioned glycolysis earlier. Picture this like a shot of nitrous oxide in a street race.
Ooh, I like that analogy.
Right. It provides a very quick burst of energy right in the fluid of the cell. It's incredibly fast, but it burns out quickly.
It's definitely not sustainable for a long haul.
Exactly. And the second power plant is the mitochondrial engine.
Right, the oxidative phosphorylation, driven by the electron transport chain. As you probably know, this is a much more complex, highly efficient machine. Electrons are harvested from our food and passed down a literal bucket brigade of protein complexes.
Like complexes I through V, right?
Exactly, moving through the TCA cycle and the transport chain, ultimately creating a massive pressure gradient that just churns out ATP.
So it's like the heavy-duty diesel engine for the cell. And here is the first major discovery from the source material. The researchers took these immune scouts and stimulated their pathogen recognition receptors.
Basically triggering the cell's radar dishes with chemical mimics.
Right. They use specific agonists like CpG to trigger TLR9, PIC for TLR3, and R848 to hit TLR7 and TLR8. And when those alarms went off, the special ops cDC1s did something totally unexpected.
They persistently increased their respiratory rates.
Yeah, both their basal and maximal respiratory rates. They heavily revved up their mitochondrial diesel engines and kept them running for a grueling pursuit.
And what is so fascinating here is that the cDC2s, that standard infantry, did not do this. Their respiration spiked briefly and then just dropped off.
Which perfectly isolates how uniquely reliant those cDC1s are on that diesel engine.
Okay, let's do a quick recap of what we just learned for you listeners.
Thank you.
First bullet point. The old dogma said mitochondrial activity meant immune suppression. This paper proves that's wrong. Second bullet. cDC1s are elite scouts that prime cancer-killing T cells. And third bullet. When cDC1s detect a threat, they constantly rev their mitochondrial diesel engines, unlike the standard cDC2s.
Spot on.
So here's a question for you to think about. Think about your own energy levels. If you were an immune cell, would you rely on a quick sugar rush, or a slow-burning mitochondrial engine to fight a tumor.
A really good framing for what comes next.
Right, because it makes you wonder, do they just prefer this diesel engine, or is it absolutely necessary for their survival? How did the researchers test that? Well, to answer that, the scientists had to physically break the engine. They used a highly specific genetic knockout model called the itgaxed mice.
Okay, that is a mouthful. What does that actually delete?
I know, it's a bit technical.
Yeah.
It specifically deletes the UCAC gene, which codes for the QPC protein.
And where is that protein located?
That protein is subunit 7th of complex 3 in the electron transport chain. So they're just pulling one specific gear out of the machine. They also verified this using pharmacological inhibitors like antimycin A and mixothiazole, which literally just jam complex 3.
Throwing a wrench into the electron bucket brigade.
So let's look at the clinical impact of that broken gear. The researchers set up a really compelling case study using a B16 OVA melanoma mouse model.
A classic cancer model, yeah.
Right. They took these break-in cDC1s, loaded them with a specific tumor antigen OVA in this case, and essentially programmed them to act as an anti-cancer vaccine. Then they injected them into mice with spreading lung melanoma. What happened?
The outcome was devastating, honestly. The impaired cDC1s completely failed at their mission. Wow. Failed how?
First, they failed to upregulate crucial co-stimulatory molecules on their surface. Specifically, the proteins CD40, CD80, and CD86.
You can think of those surface molecules as the mandatory secret handshakes required to activate the T-cell infantry.
That's exactly what they are.
Yeah.
Without those handshakes, the T-cells just ignore the warning. And not only that, they lost their navigation systems.
Right. They failed to upregulate a receptor called CCR7.
Exactly. CCR7 acts as a molecular GPS antenna detecting chemical trails that lead to the popliteal lymph nodes. Because they couldn't migrate there, they were just stranded.
They couldn't even find the T-cells to attempt the handshake. So there was a total failure to trigger the proliferation of the OT2, which are the CD4 plus helper T-cells, and the CD8 plus killer T-cells.
Exactly. And because the heavy artillery was never mobilized, the melanoma nodules in the lungs just grew completely out of control.
But wait, what about the CDC2s?
That's the amazing control in this experiment. The cDC2s were largely unaffected by this exact same complex third impairment, proving how uniquely dependent cDC1s are on this specific metabolic pathway.
Okay, time for another quick 5-minute recap. Bullet 1. Researchers broke complex third in the mitochondria of cDC1s using genetics and drugs. Bullet 2. They used these broken cells as a cancer vaccine in melanoma mice. Bullet 3. The vaccine completely failed. The cDC1s couldn't form the secret handshakes CD40, 80, and 86, and couldn't navigate to the lymph nodes using CCR7.
A complete systemic failure.
So my question for you listening, if breaking complex third ruined the cell's function, what exactly did it break? What do you think happens to a cell when its power plant goes down?
Well, most people would guess it's an energy crisis.
Right. I have to push back here. We are talking about a broken power plant. If the diesel engine is jammed, the obvious answer is that the cell simply ran out of ATP, right? Or maybe it drowned in reactive oxygen species, or ROS.
That is the most logical assumption. It's what the entire field expected. But here is the massive plot twist. The ATP and ROS levels in those broken cDC1s were completely normal.
Wait, really? How is a cell with a broken power plant maintaining normal energy levels?
Cells are incredibly adaptable. They compensated. The cDC1s ramped up glycolysis heavily to make up for the specific ATP deficit. They weren't starving, and they weren't suffering from oxidative stress.
Okay, my mind is slightly blown by that. If it has plenty of energy, but it's still failing to sound the alarm, what is the actual culprit?
To find out, they used targeted liquid chromatography mass spectrometry, or LCMS.
Which lets them meticulously weigh and measure the exact chemical metabolites inside the cell.
Right, and they discovered a profound biochemical bottleneck. Because electrons couldn't flow cleanly through complex slurry, the NAD plus to NADH ratio severely dropped.
Like a massive metabolic traffic jam on the highway.
Exactly. Raw materials had nowhere to go, so they piled up. The LCMS showed a massive accumulation of a metabolite called 2-hydroxyglutarate, or 2-HG.
2-HG, okay.
Yes, alongside a large accumulation of succinate and a notable drop in serine.
So we have this huge pileup of 2-HG and succinate. How does a pile of metabolites stop an immune cell from fighting cancer?
This brings us to epigenetics.
Let's use a mnemonic device here to help everyone remember this complex chain. Remember MET, that stands for metabolites dictate epigenetics, which dictates transcription. MET.
I love that. MET is perfect because those accumulated metabolites, specifically that high 2-HG to alpha-ketoglutarate ratio, they chemically inhibit a vital family of enzymes.
The alpha-KG-dependent methylcytosine dehydrogenases, right?
Yes, and the most important one in this story is an enzyme called TET2.
Okay, let's explain TET2. What does it normally do?
In a healthy cell, TET2 removes chemical padlocks methyl groups from your DNA. It keeps the DNA physically open and readable.
Yeah.
The 2-HG and succinate literally paralyze TET2.
So the padlocks stay on.
Exactly. The cell experiences severe DNA hypermethylation.
Let's try to visualize this. Imagine your DNA is a massive, sprawling library of instruction manuals. Your elite scouts need to access the emergency response manual instantly when they spot a cancer cell.
Right, they need it fast.
So they leave bookmarks in specific pages, so the manual is always cracked slightly open. We call this a poised state. In the cell, these bookmarks are specialized pioneer transcription factors named PU.1 and AP-1, specifically JUNB.
That is a brilliant way to explain it. In a healthy cDC1, PU.1 and AP-1 keep those immediate early stimulus-induced genes slightly open.
But when complex THR breaks and 2-HG builds up and TAT2 is paralyzed?
A heavy physical padlock, the hypermethylation is snapped onto the book right over those exact PU.1 and AP-1 bookmarks.
So the cell isn't dead, it has plenty of ATP. But when the alarms ring, it reaches for the instruction manual to build the handshakes and the GPS antennas, and the pages are locked shut.
It takes far too long to pick the lock. The signal is critically delayed, the T cells never get the message, and the cancer escapes.
It is a stunning demonstration of cause and effect. Okay, time for our third recap.
Let's do it.
Bullet 1. The broken mitochondria didn't cause an energy shortage, it caused a metabolite traffic jam. Bullet 2. Remember, MET metabolites dictate epigenetics, dictate transcription. Bullet 3. 2-HG built up, paralyzed TET2, and snapped padlocks onto the PU.1 and AP-1 bookmarks in the DNA.
Perfectly summarized.
So audience, how incredible is it that a basic metabolic traffic jam physically locks down your DNA? And more importantly, how might we bypass this roadblock?
Well, the researchers actually figured out how to bypass it, and it is one of the most elegant biological hacks I've ever seen.
The rescue mission. Tell us about the C-squirt.
Yes. To prove their theory, they used a gene from a marine invertebrate, a sea squirt called Ciona intestinalis.
I am obsessed with this detail. A C-squirt gene fixing a mammalian immune system.
It's wild. They used transgenic AOX 26 mice engineered to express an enzyme from the sea squirt called alternative oxidase, or AOX.
Because mammals don't naturally have AOX. What does it do?
AOX accepts electrons directly from coenzyme Q early in the transport chain and dumps them straight onto oxygen to make water. It completely bypasses the broken complex 3.
It's literally a bypass highway around the traffic jam.
Exactly. And the genius part, AOX does not pump protons. It makes absolutely zero ATP.
Wow. So if the immune failure was an energy problem, AOX wouldn't help at all.
Precisely. But AOX does cleanly restore the flow of electrons and the NAD+ to NADH redox balance.
And the results were triumphant. By turning on the C-squirt AOX highway, the traffic jam cleared, the 2-HG levels plummeted to normal.
And because 2-HG was gone, TET2 woke back up.
TE2 marched into the library and ripped the padlocks off the PU.1 bookmarks. The cDC1 cells could read their manuals again, and when they tested that anti-cancer vaccine in these AOX mice,
it worked perfectly. It massively reduced the spreading melanoma nodules in the lungs.
The ultimate proof of mechanism. But they didn't stop there. They brought in one final incredible piece of evidence using vitamin C.
Yes, standard L-ascorbic acid. In biochemistry, vitamin C is a well-known cofactor for the TET2 enzyme.
Meaning it supercharges it, right?
Right. They hypothesized that if paralyzed TET2 is the core problem, directly stimulating it with vitamin C should have the opposite effect.
And it did. When they applied vitamin C to healthy, wild-type dendritic cells, it drastically boosted their expression of CD40, CD80, and CD86.
They became hypervigilant. And to confirm it, they created a separate model where the TET2 gene was globally deleted.
And those TET2 knockout mice perfectly mimic the exact same catastrophic immune failures as the mice with the broken mitochondria.
If we connect this to the bigger picture, this definitively proved metabolism is upstream of epigenetics.
Okay, final recap before we wrap up. Bullet 1, scientists used the C-squirt AOX enzyme to bypass complex 3. Bullet 2, AOX restored electron flow without making ATP, proving energy wasn't the issue. Bullet 3, vitamin C, a TET2 supercharger, boosted immune function, proving the epigenetic link.
It's an airtight case.
So, listener, Could the future of treating immune deficiencies lie not in standard immune drugs, but in metabolic and epigenetic tweaks?
It absolutely could. OXPHOS is not just for immune tolerance. It is the vital engine that keeps our anti-tumor defenses poised and ready to strike.
We really traced it all today, from a microscopic disruption in electron flow to a chemical pool of metabolites to physical locks on your DNA via TET2 and PU.1. We saw how elite immune scouts get completely stripped of their weapons against cancer, and how a C-squirt or a dash of vitamin C can rewrite the code.
It completely rewrites the textbooks.
It really does. So as you go about your day, I want to leave you with a lingering thought to mull over.
I'm curious.
If something as fundamental as the metabolic flow in our mitochondria dictates the physical shape and readiness of our DNA, how much of our daily metabolic health, what we eat, our oxidative stress, our daily nutrient intake, is secretly, quietly reprogramming our immune system's frontline defenders right now?
That is a profound thing to think about.
Keep questioning the microscopic world around you, and thanks for taking the deep dive with us.